viral vectors are produced from living cells, and they're incredibly complicated to control, and they misbehave, and it could be a bad batch of raw materials. It could be the incubators, maybe some parameters is not functioning well. So it really is everything, everywhere, all at once. And I think that is why it is so important that for manufacturers in cell and gene, we need to be scientifically really astute to think about what is it that we're doing. We need to understand the fundamental biology of the modality that we're manufacturing. And these are the things that keeps me awake at night. Have we done the right thing? Have we looked at every circumstances? Are the products safe to use? Does it meet specification?
Welcome to the Smart Biotech Scientist. I'm your host, David Bruhlmann, and today we're diving into viral vector manufacturing with Lukas Chan, a molecular and cell biologist with over 20 years in cell and gene therapy development. Being the head of manufacture in UK's first clinical viral vector core at King's College London to founding CellVec, APAC's first GMP-certified specialist CDMO for viral vectors, Lukas has pioneered manufacturing infrastructure where none existed. Let's explore how viral vectors are revolutionizing patient care. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Bruhlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Lukas, to the Smart Biotech Scientist. It's good to have you on today.
Thank you, David. It's a real pleasure to be here today, and thank you for inviting me to speak.
Absolutely. Lukas, share something that you believe about bioprocess development that most people disagree with.
So this is quite a tough question, paradoxically. And I thought, since I'm going to be sharing some of my experiences on viral vectors, perhaps I'll use an example of viruses. So often when we talk about viral vectors, I do come across a lot of people coming to me thinking that isn't viral vectors same as viruses? Aren't we developing or manufacturing a really dangerous modality or vehicle? Are they the same? Should we be very afraid of using this modality? But actually, fundamentally, they are very, very different things, even though viral vectors derive from wild-type pathogenic viruses. But fundamentally, they're very different. And that's mainly because in the way viral vectors have been genetically re-engineered, basically taking apart the coding sequence to such an extent that it is unable to revert to its wild-type form and replicate. So when we talk about viral vectors, it really is not a virus anymore. It is nothing but a delivery vehicles that we call vectors.
Before we dive further into our exciting topic about viral vector manufacturing and development, Lukas, I would like you to draw us into your story because you have a remarkable journey from the Imperial College in London to establishing Asia Pacific first regulatory accredited viral vector CDMO. So what sparked your initial interest in gene therapy and what were some interesting pit stops along the way?
Sure, I can talk about that. I think it's been a real privilege. I'm still doing this. I've been doing this for quite a long time. Tracking back now, what was the eureka moment, I guess, the spark that started all of this was, I guess, when I was an undergraduate student at the time studying genetics in the UK, I came across an article from nothing but Scientific American, which is a magazine widely available around the world. And the good thing about Scientific American is that it makes it very relatable to people perhaps who's not done science before or still new to this. And I remember coming across an episode or an issue that mainly talks about gene therapy. And this is the 90s, this is mid-90s when cell and gene is still relatively new, although research has been going on for the previous decade. And the article really talks about the power and the potential of utilizing gene therapy, the approach, potentially one day to bring a cure in genetic diseases. And it cited a couple of examples, which is ADA, adenosine deaminase deficiency. So these are single gene disorders. And the other, cystic fibrosis. Again, a relatively straightforward, but very wide-ranging mutations that we've seen in CF patients. And it talks about approaches being explored to try and correct some of these mutations in humans. And I thought to myself, this is it. This is what I want to do. I want to graduate and I want to get into wherever that has an interest to do gene therapy research. And I've been fortunate to enroll in a graduate program, a doctoral program at the time in Imperial College London. I've been fortunate to be accepted into a scholarship under Charles Cattell and Bob Williamson's lab. And they turned out to be the pioneers in cystic fibrosis gene therapy and already starting some very, very early clinical programs using liposomes in an effort to address cystic fibrosis issues. And the doctoral program really involved creating viral vectors and specifically developing a suspension producer cell line at the time for viral vectors. And the other aspect is looking at tissue-specific targeting. So developing vectors that has ligand that's attached on the cell surface, hoping that it will then specifically target receptors in a certain type of cell types for specific gene transfer. And some of these projects, they were very exciting to be involved with, and we had successes along the way and other experiments that didn't turn out so well as you expect from a doctoral program. But I guess one of the development that managed to succeed was we had established at the time the first suspension cell-based retroviral stable producer cell line. So it was an extremely rewarding time, but the pitfalls were great. And I remember recalling some nights in the lab where I was sitting there watching my cell line grow in a bioreactor. 3 a.m. in the morning, there was not a soul in the building except the security guard came in, catching me falling asleep on the bench with a torch on my head. And I was thinking to myself, what on earth am I doing here? But those were some of the early sparks of my journey in cell and gene.
And what led you then to finally co-found a CDMO?
So this is a long journey. There were lots of lessons learned along the way. I guess from a doctoral student still trying to learn about cell and gene to setting up a viral vector manufacturing capability has been a long journey along the way. But it really is a sum of all the things that I've learned along the way, and I'm still learning along the way. And I guess one of the early concepts, the journey towards what I'm doing today, what I've been doing the last few years, really started at my time in London when I was involved in some pipeline and translational development. So we were developing one of the first cell therapy, gene-modified cellular therapy for hemato-oncology at the time. Again, another very exciting program, which turns out to be one of the first bench to bedside program that were approved by regulators. And it also turns out to be one of the first that uses lentivirus. And at the time, this is in the mid-2000s, again, in the journey of viral vector manufacturing, we're still going through the early phase of taking products into the clinic, in understanding GMP and its regulation when it applies to cell and gene therapy. So there was still immature guidance, immature processes and workflow. So being in an academic unit, we were in cell and gene. We would actually intimately involve in some of the developmental process itself when it comes to manufacturing. So viral vector is really a need internally to satisfy the demand to create a modality that will enable us to manufacture a gene-modified cellular therapy for the clinic. So it was accidental that I gone into viral vector manufacturing, albeit that my role in the program is to develop a new efficient gene transfer process to create this cellular therapy. So having succeeded in that process, which incidentally, we had a lot of lateral learning process in terms of regulations, GMP, clinical trials, involving in patient consent and all of that, explaining these complex modalities to the wider public, was that we had identified a major bottleneck in viral vector supply and manufacturing. And this bottleneck is not only just for us, but in the wider cell and gene community, during that time was a real issue. There were these amazing pipeline development in London and across the world, but really very little capability to take this through manufacturing in a GMP compliant, safe way into the clinic. So we had to establish a lot of these toolboxes and workflows internally. And it then morphed into become a really successful operation in itself, to become the first UK-based GMP viral vector core that we ended up not just supplying and manufacturing, developing vectors for ourselves, but we had an amazing collaboration with our network partners within London and Europe and in the wider field, not just with academic partners, but also industrial partners. So it's been a long journey. And I think that's what seeded the idea on looking at that critical bottleneck and that we really needed to expand and extend ability elsewhere.
Can you quickly explain to our listeners who might not be very familiar with cell and gene therapy, what are the fundamental difference compared to biologics? So we have already heard that the viral vector is actually not a virus, but what is different on the manufacturing side of things? Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Yes, so often there is a lot of comparison being drawn between manufacturing and modality in cell and gene, not just viral vectors. It could be gene-modified, autologous cellular therapy. So these are a complex modality. We're dealing with very complex living organisms in modified cellular therapy. These are living cells. These are complex biological system. And often in my learning journey on the early front runners in modality, bioprocess development, the monoclonal antibodies, where you have a very well-defined starting material. You have a cell line, a CHO cell line that's been altered, be it a hybridoma or gene-modified CHO cell line that expresses monoclonal antibodies that is very well understood as a modality. You have upstream process that's been developed over the last several decades to accommodate this modality that is hugely efficient in suspension culture upscaling. You have downstream technologies that's been designed to purify and capture and concentrate and to process this modality. Cell and gene, in a way, don't have any of these or have not yet had that journey in terms of technology development and maturation. And viral vectors and other cell and gene modality, at least not until in recent times where there are more cell and gene customized specific technologies now being rolled out, has been relying on repurposing, borrowing process technologies from the past in small molecules and monoclonal antibodies. And viral vectors is not dissimilar to that. We are having to look at ion exchange chromatography technologies. We're looking at bioreactor designs that's been readopted and repurposed from large-scale monoclonal antibodies. So that fundamentally is a lot more complicated and complex to handle, to control, simply because the modality we're dealing with is at times very unpredictable, very variable, and complex to handle.
Now, as you then co-founded the CDMO in Singapore, was this also mainly driven by a market need or a gap you observed, or I'd say a limitation? And also, what were some challenges you faced as you were venturing out to a completely different market, into the Asian market?
Yes, I think setting up a CDMO in any geographic location or any circumstances in itself, a very challenging undertaking in itself. Setting up a CDMO, particularly for cell and gene modality, particularly in viral vectors, and particularly in, I guess, an emerging market in APAC, makes it more challenging. I guess the fundamentals behind this is that at the time, there are market demand for viral vectors. There is a worldwide shortage of viral vectors, and to a certain extent today, there still is. It's a slightly inaccessible modality to supply. But we had the really good, successful experience, a very successful manufacturing agenda for my time in the UK. We had spent a number of years developing cell and gene, so we understood what the end user requirement is. We understood what is the viral vectors ultimately used for. And that's really important when we are developing and manufacturing a cell and gene modality. So the scientific understanding, my educational journey and understanding vector biology, which is fundamentally really important when it comes to manufacturing, is there. The market gap, I guess at the time we were going through a particularly acute time in supply, which is a good problem for manufacturer. I recall we're having backlogs of between 12 to 18 months before we can make available a manufacturing slot for a particular industry client. That is a good problem, but it's a perfect storm, I would call it. The other aspect to this is that viral vectors is off-the-shelf products. It is not a product, I guess, the complexity often discussed about autologous gene transfer and decentralized discussion surrounding where you should be putting your manufacturing site. Viral vector is actually off-the-shelf products that you can ship anywhere. So really the question then is we need to extend supply capability. There's just not enough supply around the world. And I think particularly Singapore within APAC presents really interesting opportunity. And Singapore in itself, a very mature manufacturing location. It has a number of years, through heavy institutional investment, has built up a fantastic network of biomanufacturing capability and infrastructure. And one of the fundamentals that afford is supply and logistic network with that infrastructure that comes with it. And that is really key when it comes to manufacturing. We need to be able to access critical raw materials. We need to be close to the airport. We need to have supply network to import. Singapore is a small country and imports most of its material from around the world. Other significant aspects to it is that it has a really strong advanced research institutes. They're already translational projects, fundamental cell and gene projects through very well-renowned institutes that's demonstrating good efficacy. There were already clinical trials that were going on. And last but not least, in Singapore, we have advantage of a regulatory agency that has already experience, I guess, within APAC region on compliance, on drug manufacturing, given that we have a big biomanufacturing industry within Singapore. And obviously, with the awareness that biologics manufacturing is now accommodating at the time a new modality in cell and gene. So there were lots of enthusiasm from stakeholders from across the board in supporting a new venture. So these form the early seeds in having Singapore as a potential good location to set up a capability that we have been seeing a very big bottleneck in supply, particularly in viral vectors.
So as you were building this GMP facility from scratch, which is a big challenge, what are the challenges you faced, I'd say, on the facility side, but also if you can quickly elaborate a bit on the process side? I mean, you developed a new lentivirus production platform, right?
Building a CDMO as a business in a new market comes with multiple challenges. I will break this down for easier digest purpose into several topics of consideration. So one is regulatory, one is scientific, one is to do with personnel. Obviously, the infrastructure, the equipment, the facility built itself. Being a commercial CDMO, we are talking about market access, business development, and also commercial expectations to the funders. And ultimately, how do we operate and bring the operation into a success, mature manufacturing entity? As I mentioned before, one of the fundamentals is to do with regulatory alignment. We're dealing with a new modality, a new product in cell and gene in an emerging market. So again, this comes back to our commercial strategy. How do we penetrate and access a global, regional and global market in a new entity that we set up in an unknown location? And one of the decisions that I thought would be fundamental is to ensure that the products that we manufacture and develop, first of all, we have the technical and scientific know-hows. And secondly, it is safe for clinical and commercial use. So I recall one of the very first conversations I've had when I first arrived in Singapore was actually to knock on the door of our regulators within Singapore to have a conversation with them. And I think this might contradict some of the other risk-averse approaches. And I felt in my lifetime in the cell and gene journey, often regulators are misunderstood in this regard. I felt it's really important to have that conversation early on. Of course, I didn't arrive at the door with a blank piece of paper. I already had some concept about the facility and the capability and the product line that we're trying to build. So it's to get that understanding within the regulatory agency to say, hey, this is what we're going to do. And what do you think? Are there any risks or concern that you may have setting something like this? And that became a very good starting conversation to a critical path in gaining regulatory support and our eventual certification. Now, when it comes to dealing with viral vector manufacturing, I think it's really important that we get the science and the technology right. It's a critical modality. It is not a commodity that we're manufacturing. And we really need to establish in-house capability to develop a process that is technically excellent. We're able to achieve the results that we set out. And in that journey to train some of the personnel, a lot of them has come from traditional biologics manufacturers who's never heard of cell and gene before, to train them. So we had a challenging tasks in terms of recruitment to bring in personnel from the regulatory side who has spent a lot of time dealing with biologics manufacture, the manufacturing scientists who come from biologics, and the scientists that come from advanced research institutes, perhaps has spent a number of years on the bench and had the privilege to recruit a team of scientists who were really excellent. And we had a lot of breakthroughs in our internal platform process capability establishment to really to get them to merge, to converge an understanding on our mission in setting up a viral vector manufacturing CDMO capability that is technically sound, regulatory compliant. And then that's when we may be able to challenge some market access. If we're really good in what we do, then maybe we can impart a level of confidence to our potential clients.
This is really good advice, Smart Biotech Scientists. Start interacting with regulatory bodies as early as you can. We've already covered that in other episodes. So thank you, Lucas, for reminding us again. This is a very good practice. Speaking about the manufacturing process itself, or the whole, I'd say, methodology of process development, as you were leading your clients from process development into scale up and eventually the large scale production, what were some success key factors you were observing as you were doing that to help your clients succeed?
Again, cell and gene therapy is a very complicated and complex manufacture to control and to define. And I've come across a really useful analogy that I've heard recently, which is everything, everywhere, all at once. And it is something that really clicked with me. And it's like holding a leaking bottle or container. And until you patch up all the holes all at once, something else will fall through. And fundamentally, that's how I think about cell and gene. And in my education, in terms of GMP, is a really rewarding and useful educational process. Nothing too small is worth negating when it comes to GMP and pharmaceutical manufacturing. No jobs are too small to not pay critical attention to. In naturally example, think about the shuttle program, running oil rigs, where the smallest mistake could have catastrophic consequence. And in a way, when it comes to the complex moving parts in cell and gene manufacture, it's not too dissimilar. We need to fundamentally think about what are the critical control parameters that has consequence to critical quality attributes. Fundamentally, when we look at biologics or monoclonal antibodies, there are far fewer CPPs and perhaps far fewer CQAs too. In cell and gene, we may have a similar amount of CQAs, but they are affected by a much broader range of CPPs because we're dealing with complex organisms. We're dealing with living cells. Viral vectors are produced from living cells, and they're incredibly complicated to control, and they misbehave. And it could be a bad batch of raw materials. It could be the incubators, maybe some parameters is not functioning well. So it really is everything, everywhere, all at once. And I think that is why it is so important that for manufacturers in cell and gene, we need to be scientifically really astute to think about what is it that we're doing. We need to understand the fundamental biology of the modality that we're manufacturing. And these are the things that keeps me awake at night. Have we done the right thing? Have we looked at every circumstances? Are the products safe to use? Does it meet specification?
That's part 1 with Lucas Chan, from viral vector fundamentals to pioneering CDMO development in Singapore. In part 2, we'll tackle the technical challenges of scale-up and cost reduction strategies that could transform patient access to gene therapies. Do you have process development questions? Book your free consultation at bromanconsulting.com/call to help you get started right away. Thank you very much for tuning in today. Please leave a review on the platform you found us on. Thank you very much for that, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.